Okay, so I'm at school and I've been isolating the DNA, and these are all the tools of the trade here. We have a centrifuge because we have to spin down liquids to make sure everything's at the bottom occasionally. Vortexer because when we have the cells from your scalp in the solution that's going to extract the DNA from them, we have to vortex every once in a while, basically agitate them vigorously to try to help bust up the cells and get the DNA out. Got a couple water baths. We had to incubate the vortexed cells at about 56 degrees Celsius for 10 minutes. And then we had to incubate them at 100 degrees, a boiling water bath, for about 5 minutes. The ingredients are this stuff called the master mix that I had to mix up. And the master mix has the DNA polymerase, the nucleotides we're going to use to build the new strands of DNA, and most importantly, or also important, the primers. And the primers are pieces of DNA that will be complementary to this portion of chromosome 16 known as the PV92 region that we want to copy. Here's tubes of DNA, and this is Lucas's DNA, hopefully. There are tiny hairs in there that hopefully had some skin cells. And I'm trying to get it to focus on the tube. I'd like for it to focus on the tube. Come on. Because I want you to see that there's liquid, but then there's a bunch of beads, tiny little beads at the bottom. And the tiny, and darn it, you can't see it. The tiny little beads are there to help bust up the cells as well. So the cells are incubated in this solution that has, among other things, these beads. Then you vortex it, and so the combination of the cooking and the vortexing should get the DNA out of the cells. Now, all the DNA samples, this is the thermal cycler, otherwise known as a PCR machine, that we're going to do this process known as the polymerase chain reaction in. And it has an aluminum block that's going to heat up and cool down. And here's the DNA samples. They have a color because we have added a solution that has that's the master mix, because you notice the master mix is kind of yellow. So it has the master mix in it and the DNA solutions. And they typically give the master mix a color so that you know you have added it. You can tell it's been added. So here is where we have to look up our recipe for the thermal cycler. And I've got several recipes programmed in, and here's one that says PV92. Maybe you can read that. And this is the one we're going to use. We'll click start. And you can see reaction volume, 40 microliters. In our tubes, we have 40 microliters of solution. We're going to lock them in so that the temperature keeps them nice and warm. And and now we have basically started our run. And again, this is what's known as the thermal cycler. And what it's going to do, it's going to run through these various temperatures. Right now it's at about 30, 31, 32 degrees Celsius, and it's going to heat up to 94. And it has an initial 2-minute period of 94, and then another minute. And this is the temperature at which the two DNA strands will separate. Then it goes down to 60 for a minute. This is known as the annealing phase, and that's when we attach the primers to the part of DNA that we want to copy. Then 72 for 2 minutes, that's known as the extension phase. That's when the DNA polymerase is going to build the new strands. Then we go back to 94 for a minute, 60, 72, 2 cycles, 3, etc., etc. And I think this recipe has us doing 40 cycles of this over and over, making copies of DNA. So we're going to go from a small amount of DNA and make a lot of copies of this one part of chromosome 16. This is going to take several hours, so I'm going to leave. But fortunately, our thermal cycler will then take our temperature down to 4 degrees Celsius, about the temperature of a refrigerator. You can see it's going to do that for infinity. So what's going to happen is I'm going to come by later, probably tomorrow, because this won't be done till it's almost 3 o'clock, till about 6 o'clock. So I'm just going to let it go overnight, and it'll keep it nice and cold. And then tomorrow I'll come in, and we're going to electrophoresis. We're going to load our DNA samples onto a gel and do some electrophoresis to separate the pieces of DNA that we have copied. And we'll hopefully have DNA and be able to genotype everyone. Okay? All right. Bye-bye.